Good afternoon, and welcome to the Brain and Behavior Research Foundation's Meet the Scientist monthly webinar series. I'm Dr. Jeff Borenstein, President and CEO of the Foundation, and your host for today's webinar. Today, Dr. Addy will present Investigating Neurobiology and New Treatments for Addiction, Depression, and Anxiety. The Brain and Behavior Research Foundation funds the most innovative ideas in neuroscience and psychiatry to better understand the causes and develop new ways to treat brain and behavior disorders. These disorders include addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, post-traumatic stress, and schizophrenia. Since 1987, the Foundation has awarded more than $430 million to fund more than 6,200 grants around the world. I'm delighted to introduce Dr. Addy. Dr. Addy is Associate Professor of Psychiatry and Cellular and Molecular Physiology at Yale School of Medicine. He's a member of our Scientific Council. Today's webinar will begin with Dr. Addy's presentation. This will be followed by a question and answer period. To submit your questions, please use the questions tab on the control panel on your screen. Please feel free to submit your questions at any time. Following the presentation, I'll ask as many as possible in the time allotted. And now I'm pleased to introduce Dr. Addy. the floor is yours.
Thank you so much, Jeff. I definitely appreciate that introduction, and it's wonderful to be here with all of you today on this webinar in this virtual setting, and to be able to share about some of the work that has been happening in the field and some of the work that we've been doing in my laboratory in particular, again, looking at the neurobiology or the brain biology of addiction, depression, and anxiety. And of course, we're also particularly interested in trying to understand and identify new therapeutic interventions for these mental illnesses as well. So again,
if I may interrupt for just a second, if you'd be so kind to share your screen.
Yes, let's make sure that goes through. So did it not come through the first time? Let's try it again.
No.
Okay. All right, did we get it this time?
Not really.
Okay, let's try one more ping. I was able to click on that the first time through, but we'll see.
Okay.
Are we in business?
Yep.
Excellent.
Yep. Perfect. Thank you so much.
All right. Well, thanks for your patience with the technical difficulties. I'm glad that you interrupted me for that. Definitely worthwhile. So again, what we'll be focusing on today are addiction, depression, and anxiety. And of course, these are all topics that impact us, many of us on an individual level, if we've had a personal experience or family members, friends, loved ones. But it's something that has obviously a large impact on us as individuals and as communities and as a society. But before I go into some of the work, I also did want to mention and highlight that we are also a community of individuals who are involved in this work. So I often like to start the presentations actually highlighting those in my research community who are involved in this work on a daily basis. And so what I'm doing here is actually showing some of the individuals and researchers who've been involved in this work from the past, those who are presently involved in this work. And again, a lot of this is about us working in community together to make sure that the work we do has impact in individual lives and in community and society as large. We've also been fortunate to be able to partner with several individuals to engage in community conversations. So some of the things that we do as a laboratory and as a group is to host conversations and town halls at the intersection of topics like neuroscience, mental health, faith, culture, and social justice. And so for us as scientists, we've been able to partner with psychiatrists, with psychologists, with mental health advocates, with community leaders, with faith leaders, and with those from the entertainment space and also some professional athletes who share their lived experience. And so for us, it's really been engaging and important to not only do the work in community in the laboratory setting, but to also to be engaged in the community. And in many ways, I'm grateful for this opportunity to be here with all of you this afternoon. I'm also encouraged to hear about how many people have registered for this webinar from all over the world. And I've been encouraged to see some of the questions that have come through already. So I hope that the information I present today will be informative and will be engaging. I also look forward to the questions that we'll be able to talk through at the end of the webinar as well. But again, for the purposes of today's talk, I'll be focusing primarily on the work that we've been doing within the laboratory setting, and also thinking about how that will bridge and have impact into clinical settings as well. And so one of the basic things that we're particularly interested in is what the brain basis or the biological basis is of our complex behavior as human beings. And within that framework, we're specifically thinking about behaviors that are associated with mental health challenges and mental illnesses like addiction, depression, and anxiety. So for this work, we're able to think about things not only level of human beings, but across the animal kingdom. So I'm showing a relatively old slide here, but just to illustrate something that's very obvious, that different animals have different sizes brains, as you can see here, from the smallest brain that's shown here being a mouse all the way up to larger brains like the human and the gorilla. A lot of the work that we do within research settings in my laboratory and several other laboratories starts at the level of the mouse or the rat, but with a lot of implications for humans as well. And again, we're thinking about how aspects and biology in our brain influences behavior across species. One thing I always like to highlight in this slide as well is that even though there's obviously different sizes between these brains, there are a lot of similarities as well. So when we think about specific brain structures, there are structures that are analogous in the rat and in the human. When we think about chemicals, neurotransmitters in the brain that are active and that allow us to engage in behavior, there are again similarities between the animals and humans. When we think about brain pathways and brain circuits. And so a lot of what we do within neuroscience is to try to see if we can observe certain behaviors, for instance, in a mouse or a rat, and to understand what's happening in the brain that allows that to emerge, and then to use that to understand what also facilitates our behaviors as human beings. So in that sense, we often talk about our work being translational. So taking things from preclinical animal models and applying that to humans. There are also instances when we can do something called back translation. So for instance, there may be a specific gene that's identified in a human population that's implicated in a certain disease. What researchers can do is then to take that gene, look at that specific gene in an animal model and see how manipulations of that gene may actually alter biology or alter behavior. And so that's something that's also very important. Then we can translate that back to the human, see if we can have a better understanding of the disease, and even potentially have therapeutic interventions to actually alleviate some of the symptomology associated with any specific disease. So in the context of what I'll talk about today, we're thinking both about translation up to the human, back translation back to the rat model, and vice versa, and hoping that that will give us some insight to be able to move things forward. So again, with this idea of thinking about complex behavior, we're focusing on behaviors that we associate with substance use disorders and mood disorders. Now, if we were all in the room together in a large room, I would actually take this time to ask people to start to share some ideas. In the virtual setting, I'll just demonstrate and talk about some of the ideas that people have shared over the years. But oftentimes I'll ask people to say and share ideas about specific behaviors that they associate with substance use disorders. And I've done this over the years in classroom settings, in public settings, and people are often very good about giving specific behaviors that usually reflect some of the symptoms that are associated with substance use disorder, whether that's things like taking more of a substance than a person initially intended, whether it has aspects of withdrawal, whether it's aspects about tolerance or craving, or having preoccupations with using the substance as opposed to other life responsibilities. People are very good about sharing a lot of different behaviors that are associated with substance use. And similarly, people are also good about sharing behaviors associated with mood disorders. You may be able to even think about some as we're going through this talk. And those may be behaviors that are associated with something like major depressive disorder, or it could be behaviors associated with anxiety disorders, whether that's generalized anxiety disorder or specific panic anxieties as well, or panic disorders. So again, from a research standpoint, within the lab, we're often trying to think about these behaviors and trying to look and investigate what in the brain is allowing those behaviors to emerge in the first place. And more recently, we've also started to think about the intersection and the interaction and relationship between these two. So as several of you in the audience may be familiar with, those who may be navigating or struggling with substance use disorders often have a higher prevalence or higher likelihood to also have coexisting mood disorders. So the word that we use in the field is often comorbidity. So someone may also be navigating major depressive disorder or some other type of mood disorder. We also see this vice versa. So those who are navigating through mood disorders have a higher likelihood to have a coexisting substance use disorder. So within the laboratory, we've also been thinking about whether there's some underlying common biological aspect in the brain that's allowing both of these to emerge at the same time, and whether that might also provide us with a window with which to try and treat these challenges and disorders at the same time. Now, the other question that I often like to pose, particularly when I'm speaking to a scientific audience, so if I'm speaking at a medical center or at a college, is to make a statement and see whether people agree or disagree with the state of our field in general. So the statement that I often make is this: as a scientific community, we have a clear understanding of the neurobiological basis of psychiatric illnesses. And so when I ask this question, I'll ask the audience to either agree or disagree. I've been asking this question for about 5 or 6 years, and I've never had anyone agree with this statement. So everyone has always disagreed with the statement, and people often have, in these scientific circles, often have very clear examples of why they think this statement is not true. So this is not to say that we have no understanding of the neurobiological basis, but whether we have a clear understanding is still something that people feel very strongly about. So on the one hand, people are saying we don't have a clear understanding yet of the entire neurobiological basis. On the other hand, in my title slide, I did mention that in this talk, I would be describing our understanding of things like addiction, depression, and anxiety. So there's a little bit of a tension there, obviously, in the way that I frame that. But I think it's an important point for us to wrestle with and to acknowledge. And we have made progress over the years in getting understanding about certain aspects of the neurobiological basis of psychiatric illness. And some of that I will go through in the talk as well. But at the same time, there are also areas where we still need a lot more information and a lot better knowledge. So one example that people sometimes give is that of major depression, for instance. So we do have some understanding of the things that can happen in the brain that can lead to a higher likelihood for major depression. But sometimes we may also have two individuals who both have major depression, but have very different biology that might actually be contributing to that depression. And so for us as a field, it's really trying to understand what the biology is that's contributing to different symptoms that might all fall into the category of depression, but might have two very different bases. And furthermore, even though I'm focusing on the biology as well today, or primarily, this does not negate what's happening in terms of the experiences that we have and how psychological components may also impinge or interact with our biology. And so I think that's always very important for us to acknowledge that our experiences also impact our biology and can also influence what manifests in terms of psychiatric illness. So I always like to start there and at least frame that in the sense of thinking about this in a very holistic manner, even though within the research laboratory itself, we're definitely focused on trying to understand the biological basis. And as a scientist, I always try to interact with my clinical colleagues as well, who often remind me that even though we're still trying to get a clear sense of the underlying neurobiology, we do have many effective treatments which can help people navigate with these mental health illnesses. At the same time, it's also important to acknowledge that there are areas where those treatments do not always work. So there are situations where people may have treatment resistance. And so again, we still do need much more improvement in terms of trying to develop effective therapeutic interventions for the many psychiatric illnesses that people may face at certain points of their lives. So before I go into the specific research which we have done in the laboratory, I thought it would be good to set a foundation, and I'll start by focusing primarily on the addiction component of things. And so if we're talking about substance use disorder, we of course have to start with considering how the drugs actually act in the brain. So at a very basic level, this is likely review for several of you, but did want to just start talking about neurons. And these are the cells in our brains which actually communicate with each other. Now, these aren't the only cells in our brains. There's also immune cells in our brain, which are known as glia, and they play a very active role in our normal functioning. They support the brain structure, and there's a lot of evidence showing how they are also involved in some illnesses and diseases. And so specific impacts on the glia can also impact and have influence on psychiatric illness. But for the purposes of today's talk, I'm going to be emphasizing the roles of some of these neurons and some of the pathways that we're looking at in the brain. So just by way of review, as I've shown here in the image, these neurons are composed of a cell body, which is really the energy housing of the cell. These neurons, as many of you know, have electrical and chemical properties. So just like electrical wires, they can actually have electrical signals sent down them, and that's a way that they can communicate with other neurons. The electrical signals can be sent down the axon to the area here that's shown as a terminal. And there, those electrical signals lead to the release of certain chemicals, neurotransmitters, which then can be released, can signal and actually contact other neurons, specifically at the dendrites, and contact certain receiving areas called receptors, where that process can start again. It will send a new electrical signal down the next neuron to that axon and release certain neurotransmitters. So again, when we think about drug actions in the brain, one of the principles that we often talk about is the fact that drugs are chemical ligands, meaning they can bind to these neurons, and they can interact with brain neurotransmitter systems to actually change the way that people feel. So what I'm showing on the right here is a slide and an image from the National Institute on Drug Abuse highlighting some of these properties. In this slide in particular, they are focusing on the brain reward pathway. So what's shown here in the middle is the ventral tegmental area. This is actually an area where dopamine neurons have their cell bodies. So in a sense, the dopamine neurons live in the ventral tegmental area. But the axons that I'm referring to get sent to different areas of the brain. So several areas receive these dopamine axons, but I'm just highlighting two in this slide here. One area that receives those axons is the prefrontal cortex. So this is the area of our brain that's very important for higher order thinking, for some of our executive function. It's also an area that's important for emotion control and emotional regulation. It's actually an area that's targeted in some of the new therapeutic treatments for depression, specifically some of the transcranial stimulation that stimulates this area to help with some of that emotional control. In this image, they've also highlighted the nucleus accumbens. So this is another region that receives axons from the ventral tegmental area and where dopamine can be released. This is an area that's very important for how our brain processes pleasure, and it's also important for how we have our behaviors that are directed towards certain goals. So we call that goal-directed behavior. And what's highlighted in this slide is that this area, the nucleus accumbens, is one that's particularly engaged when rewards are present. So this happens with natural rewards, thinking about things like your favorite food or music or sex. These all lead to increases in dopamine concentration within this area of the brain. Now, all substances that are abused also lead to increased dopamine in this area of the brain as well. So shown in this example is cocaine. All substances do this by different mechanisms, but the same endpoint is true for all of them, that they dramatically increase dopamine levels in this nucleus accumbens area of the brain. And they do so at a level much higher than you would see with natural rewards. So that's sometimes why people will describe having a euphoria or a buzz or a high that's very intense when they're using this substance, because it's much more than you would see with a natural reward. So again, the main point here to really emphasize is that the drugs act in the brain to lead to these changes and several others. This, of course, is a central piece, but only just one of the actions that drugs have in the brain. So by way of introduction, I did also want to highlight some of the other things that we as a research field have been able to identify over the years in terms of the different stages of addiction, as they are called. So there's a lot of different brain areas here on this slide. I'm not going to go through all of them in detail, but just want to emphasize the fact that, again, the drugs can act in these areas of the brain, and this is actually what underlies this feeling of euphoria. So again, with early drug experience, people will have a positive or reinforcing effect of the drug, or they may dislike it. They may have an aversive effect as well. Neither of those responses are learned. These are the innate responses that happen when the drug exposure is present. But eventually, over time, that can develop into a habit. And so what lots of investigators have found looking in different research labs is that there are different brain circuits and different locuses in the brain that come online when you shift from this initial exposure to drug to more of a habitual response. So oftentimes you'll hear people say that they no longer feel that same euphoria, that same buzz when they use a specific substance, but they seem to still be using the substance out of habit. And from the neurobiology, there's evidence that there's a shift in terms of which brain regions are now more active and contributing to that continued use of the substance. Another phase of the cycle of addiction, as it's referred to, is an aspect of withdrawal and negative affect. So if people are no longer using a substance, if they've abstained for a certain period of time, often there's withdrawal that comes with that. That can be a physical withdrawal. It can also be an emotional withdrawal. So in terms of physical withdrawal, there can often be physical manifestations of that in terms of how the body reacts. But people can also have emotional withdrawal as well, and it can also have negative affect, irritability, or anxiety. And from a brain standpoint, it turns out that what's happening during that time is a lot of the brain circuits and pathways that are involved in stress and in the pain circuitry are also actually activated during these periods of withdrawal and negative affect. Then another phase that people also talk about is the preoccupation, anticipation, and craving phase. And again, there's a whole host of other circuits and loci that are involved in this process as well. And so this is characterized by having a very strong drug craving that actually promotes relapse behavior. And again, there are certain pathways in the brain that are involved in this process. And so again, here just highlighting that we have gained traction in the field in terms of understanding some of the brain pathways that do contribute to these different aspects of what we associate with addiction. So for us as a laboratory, one of the things that we decided to focus on early on was this drug craving and drug relapse component, especially since this is a major barrier that prevents people from continuing to abstain from drug use if that's something that they've desired. Sometimes when we're teaching our med students, we'll actually bring somebody in who is in recovery or going through a treatment program to actually share some of those experiences with the students. And one of the things that's come up sometimes in those conversations is how intense this craving can really be. So we've had individuals who said, perhaps they've gone off to a treatment facility for a period of time, they've done well there, they've been able to abstain from drug use for quite a while. They come back to their same environment and the craving for the drug is really intense. So they describe it to the students as having a craving for your favorite food, but multiplying that by 1,000-fold. So making it that much harder to really continue to abstain from drug use when that craving is so strong. And so for us, we've been focused early on in the lab and trying to see if there are ways we can decrease that craving to help people continue to have abstinence when they're seeking to abstain from drug use. And so we were also influenced by a lot of the work in the field that had started to look at this process and how this craving emerges in the first place. So by way of example, just to pair these two things together, I did want to walk through a specific anecdote here. So let's say I'm a smoker, either someone who smokes combustible traditional cigarettes, or I'm someone who vapes. So if I started out early on in drug use, what is actually happening is what I talked about before. So just having exposure to that substance, nicotine in particular, leads to an increase in dopamine concentrations in the nucleus accumbens. This is an example I'm showing on the right from a study in rats. This was actually with cocaine, but this has been seen with nicotine as well. And again, that's associated with the buzz or the euphoria that I would get from smoking a cigarette or from vaping. So let's say I'm a person who also likes to smoke every time I go to the bar. But let's say over time I decide that I really want to quit smoking. There's all the evidence about all the other carcinogens in the cigarette. I know that there's an increased risk for cancer. And so I decide, okay, I'm going to try and quit smoking, but I still really enjoy going to the bar. So one thing that I would notice is that every time I go to the bar, I have a very intense craving for a cigarette. And so it seems like it's that much harder to quit when I'm in that specific setting. And so what we've been able to do as a research field is actually understand what is happening in terms of the underlying biology in the brain that allows that craving to occur and emerge. So again, going back to experiments that were performed in rodents, the experiment that I showed you on the top, there was also a cue that was associated with that drug delivery. And so what the experimenters found in this specific example is that if that pairing has been made over time and you just present the cue by itself, you already have this rapid increase in dopamine. So this increase in dopamine concentration used to be associated with the euphoria or the buzz of nicotine itself. Now it's associated with the cues that were tied to nicotine use, so much so that that increase in dopamine actually elicits craving and goal-directed behavior to obtain a cigarette, making it that much harder for people to quit. So one of our early focuses early on the lab was trying to see if we could dampen that response to those cues, that dopamine response, to see whether that might alleviate the craving and also make it easier for people to continue to abstain from substance use. And so there's a lot of information on this slide. What I'm really doing is just trying to highlight some of the complexity of these systems. So here again, I'm highlighting the ventral tegmental area, again, which is where the dopamine neurons live, and then highlighting the area in nucleus accumbens where dopamine can be released. And so one of the things that we thought about early on was trying to understand, again, this role of dopamine. We did find that if we increase dopamine activity, we were able to kind of increase craving and relapse behavior. If we block dopamine activity, we were able to block relapse behavior. So we started to think about specific ways that we could try to actually modulate the dopamine system and potentially have an effect on relapse behavior. For this work, we actually partnered with a collaborator, Dr. Anjali Rajadhyaksha at Weill Cornell, who's an expert on L-type calcium channels. And what she brought to our attention was the fact that these calcium channels are known to actually regulate dopamine activity. So as the name implies, it's a channel that can open and close and actually lets calcium into cells. That calcium that comes into the cell can actually change the properties of the cell and the intracellular environment, so much so that it can promote the firing of these neurons, so the electrical activity. And what I'm showing you here is some work that had shown that if you directly stimulate these specific channels, you can actually cause these neurons to discharge more electrical activity, or to fire, as we say in the field. And you can see that there's an increased rate of firing that happens here, happening much closer together, and so these cells are becoming much more active. So that was something that was very intriguing to us. There are also a few other reasons that this specific channel was of particular interest. As I mentioned, it does regulate dopamine cell activity and dopamine release in the brain. But there are also alterations in these channels that are associated with neuropsychiatric illnesses in humans. So this has been seen with schizophrenia, this has been seen with bipolar disorder, and has also been seen with cocaine use disorder. Now, I should mention these L-type calcium channels are not just in the brain, they're actually throughout the body, and they're on the heart as well. So they're also involved in regulating some of the electrical activity of the heart. And it turns out that there are FDA-approved L-type calcium channel inhibitors that are prescribed for hypertension. So this is a medication that's already available on the market. And with this increased attention to its role in neuropsychiatric illnesses, there's an opportunity to perhaps repurpose a medication that's used for hypertension and also be able to use it to address some of these neuropsychiatric illnesses. So for us, we were particularly interested in trying to understand whether these L-type calcium channels could also regulate this dopamine activity and this relapse behavior. So for that, we needed a way to be able to model and look at some of this behavior. And again, keeping in mind that we do some of our work in rats in particular with the idea of translating this to humans. So what we decided to do was use what is a classical model in the field, and that is drug self-administration. So it turns out that rats will actually take substances that humans will take as well, and they'll show a lot of similar behaviors to what we see in humans. So in this particular setup, what we have here is an operant setup where the animal is placed here with this wall in front of them. They have two levers, as is shown here, and they can press either one of them. One of these will be deemed an active lever. Every time they press this lever, they actually will get an intravenous infusion of cocaine. And we use intravenous infusion because it has very rapid access to the brain. But at the same time, they will also have a tone that will be played in the setup where they are, and a cue light that will go off at the same time. So we use this as a way to match cues with the drug delivery. If they press the other lever, which is turned inactive, there's no program consequence. And we vary which lever is active or inactive depending on the specific subject that we have. And what you can see here, this is over a 10-day period. Again, the active lever produces cocaine. You see that the animals get very good at distinguishing between the active lever and the inactive lever. And they also get very good at titrating the levels of cocaine that they are seeking out on a daily basis. So after this 10-day period, we actually take the animals through a period of forced abstinence. So this is similar to what would happen in a detoxification situation, where the animals don't have any access to the cocaine and they don't have any access to the cues. After a period of time, we'll actually place them back in the operant setup, and again, I'll let them press the levers. Except this time, every time they press the active lever, they don't get cocaine. They only get the cues, so the tone and the light, that were associated with cocaine. And we basically take a measure of the behavior over time. So this is an example of experiments anywhere from 1 day of abstinence on the left, all the way out to 45 days of abstinence. And what we see is the animals press many more times for the active lever that produces the cue compared to the inactive lever, and that's all the way throughout. And so we use this as our metric. Again, this is producing the cue. We use this as a metric of Q-induced cocaine seeking. It turns out that if they had done the training at the beginning and had the cues presented randomly, but not paired with the drug, you don't see this behavior emerge. Similarly, if the cues aren't present during this abstinence day, you don't see this behavior emerge. So it really seems to be the pairing between the cues and the drug that allows this behavior to emerge, and we use this as a metric of their seeking. People have also done other types of alterations or permutations of this paradigm. Sometimes you can present the cue beforehand and see how that influences their behavior. People also sometimes will do extinction sessions, which is similar to exposure therapy, and again, present the cue to see how it influences behavior. All of which are valid and have different applications to human models, but this is the model that we've used for our experiments up to this point. So with all of this in mind, we decided to ask that first critical question. Do these L-type calcium channels within the ventral tegmental area actually influence cocaine-seeking behavior? And so for these experiments, what we've done first, again, is to have the animals learn the cocaine self-administration paradigm. We then take them through a period of 10 days of cocaine abstinence, and then we allow them to perform the Q-induced cocaine seeking test. Now, for these initial experiments, we want to specifically know what was happening with those L-type calcium channels within the ventral tegmental area. So before any of this behavior, we actually did a surgical manipulation, which is very similar to what you would see in humans, for instance, who might be undergoing epilepsy treatment, where you can actually insert distinct probes into specific regions in the brain. This is all done under anesthesia, but I should also note there aren't any pain receptors on the surface of the brain itself, and so that aspect of it is not something that's painful. But again, we do use anesthesia and all surgical protocols to make sure that this is done in a very humane way. So with those implantations in place, we can actually directly use pharmacological agents to infuse agents that can block the L-type calcium channels within that specific brain region. And what we find when we do that is that blocking those L-type calcium channels does indeed decrease their cocaine-seeking behavior. So on the left here, what we see is animals that did not get that infusion, or they had what's known as a vehicle, so no active pharmacological agent there. They still press much higher for active levers that produce cues versus those that do not. As we increase the level of the L-type calcium channel inhibitor isradipine, we decrease their behavior. And again, this is an FDA-approved medication known as Dinocerc and Prescal. So this is something that, again, could have translational efficacy. We also want to make sure and see that this was something that was robust and not just specific to that particular inhibitor. So we used a different L-type calcium channel inhibitor and saw very similar effects. Now, in terms of translation, one of the things that we're also very interested in is making sure that we're decreasing craving specifically for cues associated with drugs and not decreasing overall craving or overall reward. So what we did was to take another subset of animals, to train them to press a lever for sucrose, take them through sucrose abstinence, and then ask, well, what if we block the L-type calcium channels? Does that impact sucrose seeking? It turns out that it didn't. So this manipulation and this intervention specifically decreased seeking for drug-related cues and not for natural reward cues. Now, we were also curious again as to what might be happening to the dopamine system. Again, we had shown, and others had shown, that if you increase that dopamine activity, you promote cocaine seeking. If you decrease it, you block it. So we thought that blocking the L-type calcium channels would actually decrease the dopamine signal. We were surprised to see the opposite. So again, when we put that L-type calcium channel inhibitor into the VTA, we actually enhanced dopamine. So here you can see at baseline, we have an increase in dopamine that occurs with stimulation, and it's greater when we block the L-type calcium channels. In the sucrose-abstinent animals that have no behavioral effect, there's also no effect on dopamine signaling. So there seems to be a correlation there. We were also wondering whether perhaps there may be a baseline difference into how much dopamine could be released in these brains after certain states. So we looked at naive animals, sucrose-abstinent animals, and cocaine-abstinent animals, and saw no difference in the maximal amount of dopamine that their brain could release. So really, it was an effect of blocking the L-type calcium channels that allowed this to emerge. So again, giving us some insights into how this may be acting in these specific brains to influence behavior. But again, always thinking about translation, from a human perspective, this isn't something that we would give to a specific brain region, but in the same way that people are taking these medications orally, so they has access to their whole body, we would want people to be taking these things orally or for systemic administration for cocaine abstinence or other types of abstinence as well. So what we decided to do next was actually to give the animals an injection where the inhibitor would have access to the whole body and to see what that did to the behavior. So very same experiment, except instead of directly infusing into a specific area of the brain, we are giving an injection of the L-type calcium channel inhibitor, and we see very similar effects. So again, we have a decrease in their cocaine-seeking behavior. If we look at dopamine, we again have an increase in dopamine, two different types of dopaminergic activity, either fast, high-frequency activity of these neurons or slow-frequency activities of neurons. Both show an increase in dopamine when we block the L-type calcium channel. So we did further studies to actually demonstrate that this increase in dopamine was actually leading to the decrease in cocaine-seeking behavior. If we blocked the effect of this dopamine, we lost the effect of this radapine. So this has been particularly exciting for us, and what we're doing next is to actually set up clinical studies to look at the same question in clinical populations. So in that instance, again, because these agents are FDA approved, we can bring people into a clinical research setting, and we can ask what happens if we give people oral L-type calcium channel inhibitors. Does it decrease their craving when they're shown drug-related cues? We can also use PET imaging to see whether that's acting through the dopamine system. So that's something that we're very excited about moving forward, to be able to understand whether these initial findings, which we found preclinically in rodents, can also apply to humans, and again, whether we can repurpose some of these FDA-approved medications to also help people who are navigating substance use disorders to potentially decrease craving for cocaine or other substances and have longer periods of abstinence. So again, getting back to the slide I showed earlier, just highlighting the importance of really trying to think about different components that may be able to impact underlying circuits and pathways in the brain that can have effects on behavior, again, that we associate with these psychiatric illnesses. So for the last half of the talk, and as I make sure we leave time for questions, I did also want to talk about some of our more recent work, again, looking at some of these same processes in the brain, but thinking about this in the context of responses to stress, thinking about anxiety, and thinking about aspects like anhedonia or a lack of pleasure. And we were intrigued and first got into this work, again, because of evidence in the literature showing that this dopamine system was particularly involved and playing a critical role in how the animals and how people respond to stress and anhedonia. And for our standpoint, we're thinking, are there things that can actually regulate this dopamine system and also influence the behavior? So we were particularly interested in a particular neurotransmitter known as acetylcholine. ACH is the abbreviation. But this neurotransmitter is known as a neuromodulator because it can modulate or affect other neurotransmitters. And so since the dopamine system was known to play a critical role in responses to stress and anhedonia, and then since acetylcholine was known to regulate dopamine, we thought it may also be regulating these stress responses. Now, granted, we weren't the first people to think about acetylcholine in this way. There's actually an acetylcholine hypothesis of depression that goes back about 40 years or so, or 50 years at this point. It was first proposed by Dr. Janowski and colleagues. And what they had noticed was actually some work that had been done about 10 years prior, where specific farmers were exposed to organophosphates that were known to increase the levels of acetylcholine in the brain. So basically the way that these pesticides worked is they would block a specific enzyme in the brain that normally breaks down acetylcholine. Because that breakdown enzyme was blocked, there was an increased level of acetylcholine in these brains, and people noticed that there were symptoms like increased depressed mood or lethargy, slowed thoughts, apathy, decreased energy, and psychomotor retardation or slowing. And so Dr. Janowski and others had hypothesized this cholinergic hypothesis of depression. And over the years, people had looked at specific regions in the brain to try and understand how they may be contributing to this potential role of acetylcholine in depression. Some of that work was done in the lab of Paul Greengard, looking specifically at acetylcholine actions in the nucleus accumbens. Some of that work was done at Yale by Marina Picciotto, looking in the hippocampus. and acetylcholine in the hippocampus' role in depression. The hippocampus is often talked about in terms of learning and memory, but also does play a role in motivation and in emotion as well. And then some of that work was done by Ron Duman, again at Yale, looking specifically within the prefrontal cortex and looking within the context of some of the rapid antidepressant effects of an agent known as scopolamine. But prior to our work, nobody had really looked at the ventral tegmental area or some of these midbrain dopamine areas to see whether acetylcholine in those regions may also play a role. And so for our first experiments, we decided to actually use the same agent that was used in humans to increase acetylcholine levels and to ask what that did to stress and anxiety related behavior. So again, for the first part of our question, we're looking specifically within the ventral tegmental area where those dopamine neurons live. And for this experiment, of course, again, when we're thinking about rodent models, we can't actually ask the rats whether they're depressed or anxious, but there are certain behaviors that we can look at to get a sense or proxy of that. One of those behaviors is the elevated plus maze. So this is a maze that's elevated about 40 centimeters off the ground. The animals are placed in the center chamber and they're allowed to transverse and explore the chamber. And it's located close enough to the ground that there are no injuries that are involved if they slip off the maze, but we also do have padding we can place there as well. The interesting thing about this paradigm, if you have an agent that produces anxiety in humans, the animals will actually spend much more time in the closed arms. So they normally like to stay in these enclosed areas. There's less light exposure, they're not out and exploring, and they spend much more time in those closed arms. If you give a pharmacological agent that decreases anxiety, that's anxiolytic, they'll actually spend much more time in the open arms. So not a direct measure of anxiety, but something that seems to correlate very well with what we see in humans. So what we decided to do as a first experiment was just to ask, what if we increase acetylcholine levels in the VTA? How does that affect their behavior? So it turns out they spend much less time in the open arms. So this is an anxiogenic or an anxiety-producing effect in this elevated plus maze. Another test that we can do is called the sucrose preference test. Not surprisingly, rats and mice do like sucrose and sweet things as humans do. And so we can use this as a way to see whether they are pleasure-seeking in any sense of the word, or whether they have lost that sense of pleasure-seeking, which we would term anhedonia. And so again, here we did the same experiment. What happens if we increase acetylcholine levels? Well, what we see is a decrease in sucrose preference. So the animals that had the agent that did not have the increase in acetylcholine showed about 80% preference for sucrose. As we increase acetylcholine levels, you see that drops down to about 50%. So this would be consistent with anhedonia, or a lack of pleasure seeking. So again, going back to this original idea, what we're proposing is that stress can actually increase acetylcholine levels in the brain, and that we can mimic that if we enhance those acetylcholine levels, and this can lead to pro-depressive, anxiety-producing, and anhedonic-like effects. Of course, therapeutically, what we're trying to do is the opposite. We want to decrease these responses, especially if someone is navigating through a stressful situation or struggling with depression. We want to see if we can relieve some of those symptoms. And so for that, we've been trying to think about what are ways we can prevent this increase in acetylcholine from having its effects. And so for that, we've actually focused on one of the receptors that's activated by acetylcholine. This is termed the M5 muscarinic receptor. We've partnered with some colleagues at Vanderbilt University who developed some pharmacological agents to target these specific receptors. So I will say that we have seen that we've been able to reverse some of the behaviors I just showed you in the elevated plus maze and the sucrose preference test if we give an agent that blocks these M5 receptors. But we've also been thinking more broadly about the symptomology associated with mood disorders, and we've also been interested in this aspect of motivation or amotivation. Some of the examples that people often give when I talk about major depressive disorder is someone who might have lost some of their motivation for some of the general day-to-day activities that you used to be excited about. So the nice thing about the amotivation is that there are models that have been developed in animals that also translate to humans. So in terms of looking at motivation, one of the behaviors that we use in the lab is called the effort-related choice behavior. Basically, again, the animal is placed in the operant setup and they have access to two choices. They can either take this freely available chow and eat as much as they want. It's a normal chow that they actually enjoy. But they also have the option to have a very highly palatable pellet that they prefer over the chow. In order to get the pellet, however, they have to press this lever 5 times just to receive one pellet. So what you can do is train the animals and ask them which ones they'll prefer. So if the animals get trained, they typically take a fair amount of the freely available chow, but every once in a while they also press the lever 5 times to get them more preferred reward. This is a technique and a method that was developed by John Salomon at the University of Connecticut. And what he found is that changing the dopamine system can actually change the behavior. So if you enhance dopamine activity in the brain, the animals will actually spend much more time working for the highly preferred chow. If you block the dopamine system or deplete dopamine, they'll actually not spend much time working for the high effort reward, but just take the freely available chow. And as I mentioned, there's a human correlate of this test as well. So the human effort expenditure reward task, or EFER task, is something that was developed by Michael Treadway and colleagues. And what they found is that those individuals, humans in this instance, who show anhedonia, show a decreased sensitivity for high effort reward. Now in this task, the individuals can either use their dominant pointer finger to obtain a pretty low monetary reward, or they can use their non-dominant pinky to press a clicker several more times to get a higher monetary reward. And what he found is that individuals navigating major depressive disorder actually won't spend as much time on the high effort reward. They'll take the easier reward. And similar to the rats, if you enhance the dopamine system, you actually will increase people's willingness to work for the high effort reward. So now we have a paradigm that works both in rodents and in humans, where we can ask a similar question about motivation. And again, we've been able to use that to say, okay, well, how does increasing acetylcholine tone in the brain impact the effort choice behavior? And if we block some of these specific receptors, can we actually reverse that? So I'm first showing you the data from females. This is relatively recent data that we've collected in rats. In this first panel here, I'm just showing you how many times or how much of the chow that the animals are consuming. Here is where we increase acetylcholine levels in the brain. It doesn't really change the amount of chow that they take. Here, if we block those M5 receptors, again, it doesn't change the amount of chow. And if we do both together, increase acetylcholine and block the receptors, no change. Something very different happened with the lever pressing though. So again, this is a lever pressing for the high effort reward. If we increase acetylcholine in the brain, they don't press that lever nearly as much, so they decrease how much high effort choice they will make. If we block those M5 receptors, it has no effect. But if we increase acetylcholine tone and block those M5 receptors, we can restore the effort behavior. So their motivation has seemed to return. And so from a therapeutic standpoint, if you're thinking about individuals who have lost motivation, it suggests that blocking these M5s may actually help people regain some of that motivation. And looking in males, we see something very similar, no effect on the chow consumption. If we look at the lever pressing, again, if we increase acetylcholine, we have a decrease in their effort for the high effort choice. No effect if we block the M5 receptor. But if we increase acetylcholine and block the M5, we again have a reversal of that. We restore the motivation, and we actually restore it much higher than it was initially. So two intriguing things here, the fact that this agent had an effect both in males and females, but also that there was a differential effect. It seemed to be a much higher reversal or above baseline in the males. And so for us moving forward, what we need to do is to actually continue to look at different concentrations to see whether there's a concentration that may be most effective in females and in males. And this is a really important point, because if we're thinking about translation, we need to make sure that we're looking both in males and females, so we don't take something from only one sex and apply that to the other when there may be a different dose that's more therapeutically effective. And as a last two slides before I open it up to questions, did want to just highlight some of the work we've been doing, again, thinking about comorbid or coexisting substance use and mood disorders. So with these, we've been using the elevated plus maze to investigate this question. Again, as I showed before, we found that certain manipulations that decrease or that alter dopamine signaling can actually decrease cocaine-seeking behavior. And it turns out that during cocaine abstinence, these animals actually show a heightened sense of anxiety. So here, these are animals that were undergoing saline administration. You see they still spend about 50 seconds in the open arm. If they're in cocaine abstinence, they basically do not venture out of the closed arm whatsoever. So they have a very pro-anxiety type of phenotype, which we can reverse if we use the same manipulation that decreased cocaine seeking. So now we have a situation where we may be able to decrease craving and actually reverse the anxiety that exists during the abstinence period simultaneously in the same individual. And so that also has therapeutic implications for situations where people may be struggling with substance use disorder, and may also have negative affect or anxiety at the same time. And we've also seen very similar effects if we block the L-type calcium channels. So no effect in animals that did not take cocaine, but we can reverse the effects of cocaine abstinence if we block those L-type calcium channels. And as a last slide, we've also looked at the effects of chronic stress as well. So chronic stress can also lead to pro-depressive effects in humans. We see that in our animal models as well. And basically what I'm showing you here is that we do indeed have a decrease in sucrose preference after chronic stress. If we give a systemic injection of that L-type calcium channel antagonist or inhibitor, we can reverse that. And so now you have no effect of the chronic stress. We see that in males. In females, there's a trend towards that, not significant, but again, we need to think about different doses that we may need to use between males and females to see what is most effective. And again, we're excited about moving some of these things forward into clinical research studies as well. And we can do the same thing with our M5 studies where we can actually use those agents and try and set up some clinical studies to see whether blocking that M5 also has effects on motivation in humans as well. So with that, I will get to the most important slide, which is thanking all the individuals, again, who've been involved in this work over the years, both current lab members and alumni of the lab. And some of the collaborators I've highlighted in yellow have collaborated on the specific projects that I talked about today. But I will stop there for now and leave it open to any questions.
Nii, thank you so much for an outstanding presentation and the work that you're doing. You really did a great job sort of bringing some of the basic science to the translational and where that goes. So thank you for a great presentation. I want to start off, you made reference to transcranial magnetic stimulation. And as we know, it's been approved by the FDA for depression, some other disorders, but I know that work is being done on it related to chemical dependency as well. I want to ask if you could say a few words about that.
Yeah, that's a great question. And actually, let me see. This was not planned whatsoever, but
Okay.
So in a similar way, a lot of that work, again, has started in the animal models and is being translated into humans as well. So this is just showing a cartoon image of some of that. And it's actually, again, targeting some of the same brain regions that are targeted for depression. So again, these areas that are important for emotional control. And so some of that initial work was done in rodents using a methodology known as optogenetics that can activate specific areas of the brain using light, and showing that if you activate some of those regions in the prefrontal cortex, you can actually decrease responses in terms of craving or other types of addiction-related behavior. And so there are pushes as well to try and use that transmagnetic stimulation to also do that work in humans to decrease craving. I'm not sure where that is in the FDA process, but I do know that's a very active area of research among clinical researchers. I have colleagues who've been engaged in that work, and it does seem to have good efficacy from the data that I've seen so far.
Good, thank you. If the next question I ask, you have a slide for that one, everybody's going to know that we planned it in advance. I want to ask you a little bit more about coexisting comorbid dual diagnosis. It's referred in a number of ways, and about the importance of really treating both at the same time. For people out there who may have substance abuse and also a depression along with it, could you speak a little bit about the best approach that they should take for help?
Yeah, so I think the very first thing that comes to mind, and oftentimes in our community conversations, is just the acknowledgement that both of those exist. I think sometimes there is a tendency to only focus on one or the other. And as you alluded to, that can make it difficult in some ways as well. So speaking first from just a very basic science standpoint, if we are giving agents that might decrease the craving, but there's a heightened sense of anxiety that's causing disturbances and pain, then that also may end up leading to relapse. And so these things do intersect and overlap. And so I think the first step is having that acknowledgement. But then also when people are considering which ways to approach this, I know there were some questions also about trying to have interventions that do not involve medication. So medication is one approach. It doesn't always have to be the approach, but I think whatever that approach is, it needs to be something that takes into account both of these components. So whoever that provider is, whether they're willing to work on both those components or be part of a therapeutic team that will acknowledge both of those components, I think it's critical, because otherwise I think we're not helping people as fully as we can, because we're not taking into account all the different situations and challenges that may be there.
Good, thank you. I want to ask you to speak a little bit about stigma for both depression and other psychiatric illnesses, and for chemical dependency, and how that gets in the way of people seeking treatment and really getting engaged in treatment.
Yeah, that's an excellent question. And so you're exactly right. It does get in the way because it prevents people sometimes from having that acknowledgement of what needs to move forward. I can give a couple of anecdotes and different ways that that stigma can come out. So I have been able to be involved in these conversations of faith communities as well. And so sometimes there's a stigma there that someone has either is having a moral failure or is having a shortcoming. And so that allows the challenge to be left to the side. Sometimes it's also, people have erroneously said that it's because of a lack of faith. So maybe someone is told that they're still struggling with their depression and anxiety because they aren't praying hard enough. So the challenge there is then they are trying to pray more, which there is lots of evidence. And as someone from a faith background, I can definitely say that there's an importance there as well. But then we're not always availing ourselves to a lot of these interventions, whether psychological or medication interventions that can help as well. At the same time, I've also seen stigma in another sense, where sometimes people may be taking medication, but maybe looked down upon for that, or maybe trying to take medication, also think about being involved in communities, faith communities and other communities, and being told that they shouldn't pay any attention to those things, despite the fact that lots of evidence shows that being in community can also help people move through and navigate those circumstances. So I think on both sides, we have to really do what we can to actively diminish that stigma, think about ourselves as whole beings, and think about what we can do to actually move through and make sure we surround ourselves, if we can, with people who will support us in that journey. And then for those who have been supported in that journey, to be able to speak out against that and speak about the importance of moving through that and how stigma can prevent us from getting to that better place, and how moving through that stigma can really be very empowering and bring a pace of healing as well.
Great response, really very important information. One last question, sort of the next step in terms of getting people into treatment. Often family members may see that their loved one may have chemical dependency and may have depression, and it's hard to get their loved one to seek help. What do you suggest to people when they're faced with that difficult situation?
Yeah, so the first thing that I suggest, and a lot of this comes from, again, my clinical colleagues and some that I've been able to interact with in different ways, is just being able to be available and to be able to listen to that person and ask them, you know, what they feel like may be happening or where they feel like they need support, rather than only telling them what you think they need to do. Oftentimes it becomes a matter of knowing when people are ready to take that next step, but being available to listen and provide support can go a long way. I would also say that there's lots of resources that people can take advantage of as well. The National Alliance on Mental Illness, NAMI, is a great one, because they have a lot of resources that specifically help people navigate how to have some of these conversations with loved ones, or even with individuals who then have decided to take that next step and to seek treatment. There still can be challenges along the way, so they have support groups that can help people know how to support their family members and loved ones. But again, just to emphasize, being ready to be available to listen and provide support goes a long way, and oftentimes can eventually lead to an opportunity to provide guidance. Once that person knows that you're in their corner, you're listening to them, and you're not there just to judge them.
Excellent advice. And I think a key point along those lines is never give up. Always be there for the person to try to help them get the help that could make a difference for them.
I agree.
Nii, I want to thank you again for an outstanding presentation, and more importantly for the ongoing work that you're doing. And we're going to have you come back here to tell us some of the results of this translational work that you're doing, because I think it's very exciting and very promising. So thank you for all that you're doing.
Thank you so much.
Thank you. I also want to thank everybody who joined us today. 100% of all contributions for research are invested in our grants to scientists. All the research we made fund is made possible through through supporters like you. So please consider making a gift by visiting bbrfoundation.org or call us at 1-800-829-8289. This webinar has been recorded. If you've missed any portion or would like to share it with family and friends, please visit the events and webinar page on our website. I hope you'll join us again next month on March 8th at 2 p.m. Eastern Time, when Dr. Jennifer Dwyer, Assistant Professor, Co-Director, and Co-Founder of the Pediatric Depression Clinic at Yale School of Medicine, will present Rapid-Acting Treatments for Pediatric Depression and Suicidality. Where are we now? Thank you, and remember, together we can dramatically improve the lives of those living with mental illness and enable more people to live full, happy, and productive lives. Thank you.